Natalia Posada. I'm a medical doctor specialized in human reproduction at the Instituto de Fertilidad Humana, INSER, in Medellín, Colombia. It's a pleasure for me to talk about a very interesting topic, preimplantation genetic testing. As its name implies it, we are referring to the analysis of either oocytes, fertilized oocytes, or embryos before they are transferred to the uterus. So when we analyze oocytes or zygotes, we are actually doing an indirect analysis through the biopsy of the polar bodies. In the case of cleavage stage embryos, we usually extract 1 or 2 cells from a more or less 8-cell embryo. And in the case of blastocysts, we usually take out about 5 to 10 cells from the outermost layer of these day 5 to 7 embryos called the trophectoderm that will give origin to what we know as the placenta. Because the number of cells taken from blastocysts not only do not come from the embryo proper, but because the percentage of analyzed cells is less compared to the total amount of cells that a blastocyst contains, it is believed today that blastocyst biopsy is less detrimental to the embryo than biopsies on day 3. And the reason to do this testing is to transfer chromosomally normal embryos to the endometrial cavity in order to improve the outcome of IVF procedures. And the reason being is that human embryos have a high percentage of chromosomal abnormalities, most of which are originated in the eggs during meiosis 1 due to segregation errors during meiosis of either non-disjunction or premature separation of homologous chromosomes or sister chromatids. Regardless of the age of women, even in their peak fertility years between 20 and 32 years of age, up to 20% of oocytes carry chromosomal aberrations. So we've been trying to analyze human embryos since the 1990s, initially from cleavage stage embryos and also the polar bodies of either oocytes or fertilized oocytes, initially by genetic techniques such as PCR, and later by fluorescent in situ hybridization or FISH, initially to analyze the X and Y chromosomes in cases where the mothers carried an X-linked disease. FISH PGT was later expanded to include other autosomes such as chromosome 13, 18, and 21, in addition to the X and Y chromosomes. And ultimately, they were able to analyze up to 12 chromosomes. However, the results of RCTs published in the following 2 decades were very controversial. And finally, the results of the meta-analysis by Mastenbroek and colleagues published in 2011, which showed that this kind of PGT done on 12 chromosomes had no benefit, regardless of the subgroup of patients analyzed, gave set to the development of a 24 chromosome analysis in preimplantation embryos. So today we'll be discussing PGTA 2.0, that's how it's called today, or comprehensive chromosome analysis. We're going to overview the different kinds of chromosomal abnormalities that can be identified in embryos. We'll see the clinical applicability of this technique nowadays. And finally, we'll talk about a new kind of PGTA called non-invasive PGTA because it is done not inside the embryo, but from the culture media where the embryos actually grow in. So in order to be able to analyze all 24 chromosomes, the DNA that is extracted from the little amount of DNA that is extracted from the embryo cells needs to be expanded. And the most commonly used technique today is called whole genome amplification, or WGA. This amplified material is then analyzed to reveal the regions of the genome that are under or over represented by methods such as comparative genome hybridization, or CGH, or CGH by arrays, or single nucleotide polymorphisms arrays, and lately next generation sequencing, or NGS. Today, the combination of wide genome amplification based next generation sequences is the most popular method currently used because of its high resolution and possibilities given by the technique. So the resulting data is then analyzed in order to produce a karyotype that will encompass all 24 chromosomes with little depth, but enough to be able to analyze all 24 chromosomes. These karyotype is then aligned against the reference human genome and grouped together into bins or spaces spread out over the chromosomes. So the karyotype profile indicates the copy number of DNA for each bin, which can be uniform along a chromosome or variable, depending on the presence of chromosomal abnormalities. So we call a cell to be euploid when it has two sets of 23 chromosomes, in other words, 46 chromosomes. Whenever there is a numerical abnormality that affects homogeneously a chromosome, it's called an aneuploidy, and it's a deviation in the chromosome copy number from multiples of 23. Aneuploidies are the most common embryonic genetic abnormality, the main cause of failed implantation, pregnancy loss, and congenital birth defects. And they can affect one chromosome or numerous chromosomes within a cell. In this case, they are called complex aneuploidies. And so, depending on the number of chromosomes that are affected, one may refer to a nullosomy, where there's only 44 chromosomes instead of 46, so we're missing a pair of chromosomes. Monosomy, of which 45X0, for example, it's quite known. In that case, we're missing just one chromosome. Or we can have extra chromosomes, and that's referred to as polysomy. For example, trisomies are cases where there are 47 chromosomes, and tetrasomies, there's 2 extra chromosomes, or 48 chromosomes total. Overall, it is believed that around 50% of all embryos that are preimplantation embryos that are generated in the lab are affected by aneuploidies. So it's a very, very common abnormality in human embryos. And it is related to maternal age in a very clear and direct way. So when women are between the ages of 22 and 30 years of age, one would expect 1 in 4 embryos to be affected. But by the age of 35, around 40% of all embryos are aneuploid. And when a woman reaches the age of 40, 60% of embryos are affected. And by age of 45, almost 90% of embryos will be aneuploid. And that is probably the main reason why fertility declines steadily after the age of 35. In addition to maternal age, it seems that environmental factors may also affect the incidence of aneuploidy, and this may be related to assisted reproduction factors or lifestyle condition of parents, such as obesity, smoking, exposure to radiation. Contraceptive use might have a protective effect. So, aneuploidies affect most commonly small chromosomes, such as chromosome 15, 16, 21, and 22, and less commonly the large autosomes, such as chromosomes 1 to 6. Most autosomal monosomies and most autosomal trisomies are embryonic lethal, except trisomy 13, 18, and 21, which may lead to the birth of babies with Patau's, Edwards, or Down's syndrome, respectively. That's the reason why there's great interest in, regardless of which chromosomes are analyzed, to always include these three chromosomes when we study aneuploidies. When we talk about sex chromosomes, the clinical implications will depend on the copy number variation and the chromosome affected. In other words, an X chromosome is absolutely essential for life. In the case of babies with 47XXX trisomy or 47XYY trisomy, one can have phenotypically normal females and males. In the case of monosomies, as I said before, 45X0 may give rise to Turner syndrome, which it's one can see quite often, and or 47XYY XXY is a trisomy that may give rise to Klinefelter syndrome. When there's an absence of a whole set of chromosomes, we refer to as haploidy, and when there's an extra set of chromosomes, we refer to as polyploidy. In this case, to be exact, we are not referring to aneuploidies because we still conserve multiples of 23. However, these chromosomal aberrations are not compatible with human life. The second most common abnormality is chromosomal mosaicisms, and it refers to the presence of cells with two or more chromosomal constitutions within an embryo. In other words, some of the embryonic cells may be euploid and some may be aneuploid. And mosaicism, different from a whole chromosome aneuploidy that affects all cells within an embryo, originates during mitosis. In other words, once the egg has been fertilized and it begins to divide its cells, one cell may have a mitotic event, either due to a non-disjunction of a sister chromatid or to an anaphase lagging of one of the sister chromatids. And so the daughter cells of this cell who had the mitotic event will be affected by aneuploidy, but not the rest of the cells within the embryo. Mosaicism, since it's originated during mitosis, is not affected by maternal age, but again, there seems to be a variable incidence of mosaicism between fertility clinics. What is important to understand is that mosaic preimplantation embryos can result in successful pregnancies and healthy newborns, with lower rates compared to euploid embryos and with higher miscarriage rates. And this has come from the analysis of more than 800 transfer of mosaic embryos in the last 5 years. It seems that there's a significant correlation between the clinical outcome and the level of mosaicism, with embryos having with less mosaicism, under 50%, doing much better than embryos who have higher rates of mosaicism, as well as the type of mosaicism. Segmental mosaic embryos perform better than whole chromosome mosaics. And the complex mosaics in which one embryo can have different chromosomal abnormalities in a mosaic fashion do worst of all. In any case, When there's going to be the transfer of mosaic embryos, this should always be accompanied by patient counseling with emphasis on the need for prenatal testing with amniocentesis, as we are still evaluating what the exact risks for these fetuses will be. The third type of chromosomal abnormalities are segmental. In other words, only a portion of a chromosome is affected by either a regional loss or gain of DNA. These segmental copy number variants are due to faulty corrections of chromosome double-strand breaks that occur either during meiosis, but most commonly, 70% of cases occur during mitosis. Therefore, we can have mosaic segmental abnormalities. They affect between 6 to 15% of blastocysts created in the ART lab, account for 6% of clinical miscarriages, but only affect 0.05% of newborns. However, the syndromes that they produce are very well characterized and include syndromes such as Cri-du-Chat, where there's a terminal deletion in the short arm of chromosome 5, or the Charcot-Marie-Tooth disease, type 1A, in which there is a duplication in the short arm of chromosome 12. The question is whether embryos with segmental abnormalities should be considered for transfer. There is not as much data as there is for mosaic embryos. However, doing a second trophectoderm biopsy may facilitate the clinical decision, since there's only a 50% concordance with a second trophectoderm biopsy or when the remaining of the embryo is analyzed. And the reason being that around 70% of segmental abnormalities are mitotic in origin and therefore mosaic. So not all cells in an embryo are affected by these segmental abnormalities. Finally, we have structural rearrangements, which also are due to abnormal correction of chromosome breakages, in which there's a subsequent reunion of the fragment but in a different configuration that changes the natural order of the chromosome segments. So we may have balanced or unbalanced structural rearrangements. Examples of these are balanced translocations and inversions, which are, as the name says, usually balanced. Others are insertions, chromosome rings, or Robertsonian translocations, which are usually not balanced. Anyway, parents who are carrier of these structural rearrangements are usually asymptomatic, and they have an unaltered copy number, that is, they are balanced. However, their gametes are often not balanced due to the process of recombination and segregation that occurs during meiosis. The genetic test that is used for structural rearrangements is called PGTSR, not PGTA, because it's a targeted test when the chromosomal abnormalities present in the parental genomes are known. So the newer PGTSR platforms analyze the sequences or genetic markers in or around the breakpoint regions and may now differentiate not only unbalanced but also balanced and normal embryos, so one can transfer embryos that are not carriers so as not to impose the same problem to these children in the future. So what one sees in an NGS profile is that most chromosomes should be aligned with a 2-copy number, okay? As we can see on the y-axis. If there's a chromosome with a 3-copy number, as is the case here for chromosome 7, then we know we have a trisomy of chromosome 7. And here we have chromosome 12 that shows an alignment between 2 and 3. And this is how mosaic embryos present. Here we have a 50% mosaicism for chromosome 12, which indicates that if, for example, we had biopsy 10 cells from the trophectoderm, 5 were aneuploid and 5 were euploid. Here we can see the difference between the NGS profile and the previous comparative genome hybridization profile by arrays, in which it is obvious that mosaicism is not clearly identified as it is with the NGS profile. It's different to tell whether this chromosome 12 in the lower ACGH profile is actually a mosaic it relates to mosaicism or if it's just technical noise. And the same occurs with segmental imbalances, in which NGS profiles have high resolution and can easily detect either gains or losses of chromosomes, as we can see here for chromosome 16 and 18. However, with the comparative genome hybridization arrays profile, it is not quite obvious whether we are in the presence of a segmental loss or whether it is just technical noise. The most common referral patients categories for PGTA are advanced maternal age, that is women over the age of 35, prior recurrent pregnancy loss, prior recurrent implantation failure after in vitro fertilization, and cases where there's a severe male factor in the couple. However, other clinics just decide to do genetic embryo testing in every single embryo. So it is important to define what is the real clinical applicability of PGT for chromosomal abnormalities in order to define the populations that will benefit the most. Clinical applicability will depend on three main points. The first, it has to be accurate and results have to be consistent or reproducible, and that happens to be the case for the PGTA 2.0, that has almost 100% reproducibility. There also has to be a good genetic concordance between the results of the trophectoderm biopsy and the rest of the embryo, or at least the internal cellular mass within the blastocyst, that is where the embryos will actually be produced. And finally, there must be good clinical results after the transfer of embryos that have undergone 24 chromosome analyses, that is good implantation, live births, and low miscarriages rates. So there have been a couple of studies evaluating the ploidy or copy number concordance between trophectoderm and internal cellular mass. And for whole aneuploidies, this concordance is usually very, very high, between 93 and 100%. However, when we have mosaic results, the concordance is not as good. This mosaicism concordance is only between 25 and 29%. And for segmental aneuploidies, it's variable between 50 and 100%. And this is very important because this is the reason why sometimes mosaic embryos are transferred, giving rise to healthy babies, and the reason why we are still unsecure of how to deal with embryos that show segmental aneuploidies. In the case of karyotype concordance or exact chromosome affected concordance, this is still high for whole aneuploidies. For mosaicism, it's still around 30%, and for segmental aneuploidies, it's around 50 to 30%. So not as good a concordance for mosaicism and segmental aneuploidies. And when we evaluate the concordance between trophectoderm and the whole embryos that are donated for research purposes, again, the ploidy concordances for whole aneuploidies are above 90%, segmental aneuploidies with around 50%, and mosaicisms around 27%, and karyotype concordance pretty much the same. And what are the results after the transfer of chromosome? I'm sorry, embryos that have been analyzed through PGTA. I would like to discuss with you today two meta-analyses and three recent RCTs that evaluated the impact of PGTA in advanced maternal age women and also in good prognosis patients. The meta-analysis of Dadu and colleagues was published in 2015. It included three RCTs that were published between 2012 and 2013 that included around 700 embryos that were biopsied on day 5 or 6 and that were analyzed by comparative genome hybridization arrays or qPCR and had minimal heterogeneity. And also, they did a meta-analysis and 8 cohort studies with almost 3,000 zygotes or embryos analyzed. However, these studies were very heterogeneous. Some included polar body biopsies, other day 3 or day 5 biopsies, and were analyzed by either qPCR or ACGH. However, the results were quite consistent. Um, uh, Both the meta-analysis of the three RCTs showed a relative risk of 1.29 for clinical implantation rates and 1.39 for sustained implantation rates over 20 weeks of gestation, which were clinically significant. And the observational studies, a relative risk of 1.78 for clinical implantation rates and 1.75 for sustained implantation rates, which was also clinical significant, despite very high heterogeneity in the observational studies. The results of the meta-analysis by Chen and colleagues, also published in 2015, are quite similar. It included the same three RCTs plus an RCT by Schoolcraft and colleagues, and 7 observational studies, one of which did use wide genome amplification in NGS, and most were done on blastocyst biopsies, but 3 did include cleavage stage embryos. In other words, again, quite heterogeneous for the observational studies. The transfer of euploid embryos was associated with a higher implantation rate for the 4 RCTs, with a 30% increase in implantation rates, and a 74 increase in the implantation rates for the observational studies, results which were very similar to what was published in the previous meta-analysis. The live birth rates for 1 RCT showed a relative risk of 1.26, which was significant, and for 3 observational studies, a relative risk of 1.35, in other words, a 35% increase in live birth rates. However, the confidence intervals were not significant, and there was high heterogeneity in this meta-analysis. With respect to multiple pregnancy rates, both an RCT showed a 98% reduction in multiple pregnancy rates, and 2 observational studies, a reduction, a 81% reduction in multiple pregnancy rates, which were both statistically significant with no heterogeneity. Miscarriage rates were also reduced by euploid embryo transfer. However, these were not significant for 2 RCTs, and were very significant for 5 observational studies, with a 69% reduction in miscarriage rates and very low heterogeneity. The RCT, the next 2 studies that we're going to be looking at evaluated the impact of the use of PGTA on advanced maternal age women. The first study, called the ESTEEM study or the ESHRE study into the evaluation of oocyte euploidy by microarray analysis, basically evaluated the first and second polar body by array CGH to select embryos for transfer and see whether these would increase the live birth rate within 1 year in women with advanced maternal age between 36 and 40 years of age compared to ICSI without chromosome analysis. This was a multi-center trial with an intention to treat analysis that included almost 400 women in 9 centers between 2012 and 2016. And the reason why they wanted to evaluate the polar bodies is because in some countries, the embryo biopsy as such is not allowed. The intention to treat analysis showed there was no difference in the cumulative live birth rates per patient. It was 24% in the comprehensive chromosome screening group versus 24% in the control group, despite significantly fewer embryo transfer in the comprehensive chromosome study group. And this was due, in other words, the patients in the study group achieved the same cumulative live birth rates than the control group despite having fewer embryo transfers because they had lower miscarriage rates, 7 versus 14%, and significantly higher live birth rates per euploid embryo transfer, 33% compared to 70 to 18%, which was clinically significant. A similar study was published by Rubio and colleagues in 2017. In this case, they used ACGH analysis on cleavage stage embryos and analyzed 100 advanced maternal age women between 38 and 41 years of age who had at least 5 mature oocytes at retrieval and compared them to 105 advanced maternal age patients in the control group who only had morphology assessment of blastocysts before their transfer. Compared to the control group, the PGTA group had significantly lower miscarriage rates, 39 versus 2.7, significantly increased delivery rates per first fresh blastocyst transfer, 24 versus 53%, with an OR of 3.2, and decreased percentage of cycles reaching embryo transfer, 90 versus 68%. In other words, same results as the ESTEEM study. Again, the delivery rates per recruited patients after 6 months by intention to treat analysis were similar between the two groups, 16.1 versus 26.4% respectively, which was borderline significant. However, the patients in the study group require less embryo transfer to achieve a live birth rate, about half, and the time to pregnancy was also diminished almost to half. The last RCT I would like to share with you was a very awaited study called the STAR study, or single embryo transfer of euploid embryos, that wanted to evaluate whether NGS-based PGTA increased ongoing pregnancy rates and live birth rates after the transfer of a single vitrified and warm euploid blastocyst compared with embryo selection by morphology alone in good prognosis patients. These were not advanced maternal age patients. These patients' ages were between the 25 and 40 years of age and had to have at least 2 blastocysts available in order to be randomized. 32 clinics participated in this study with a total of 665 women. Again, there was no difference in the intention to treat analysis, but different to the previous studies, there was also no difference in the ongoing pregnancy rates per embryo transfer, which was 50 versus 46%, both of them very high percentages, or per, as I said, intention to treat analysis at randomization, 41 versus 43%. A post-study analysis, however, did show a significant increase in the ongoing pregnancy rates per embryo transfer in a subgroup of patients between 35 and 40 years of age, which was 51 versus 37%, which was significant. This, however, was not evident by an intention to treat analysis, and it's possibly due to the fact that in 12% of patients with PGTA who did not have an embryo transfer, 60% did have at least one mosaic embryo, which nowadays would have been transferred and which would have probably increased the pregnancy rate per intention to treat. So, which studies really do benefit from PGTA analysis in a cost-effective manner? It seems for now that advanced maternal age women seem to be, better indications than good prognosis patients. However, there's have been two studies that have evaluated the cost effectiveness of PGTA according to the women's age and the number of available blastocysts, comparing a conventional strategy where all available blastocysts from one IVF stimulation cycle are transferred sequentially. The first in a single fresh embryo transfer, and then in case there's no pregnancy, they will be subsequently thought for selective embryo transfer of thought one blastocyst at a time, versus a systematic use of genetic testing of the trophectoderm in blastocyst stage embryos, which are all frozen, and then only euploid single embryo transfers are sequentially done until all embryos are used or the patient has a delivery. And the first cost effectiveness analysis by Somigliana and all published in 2019 was a theoretical cost-effective analysis based on the costs of the regional public health system. So what they were able to see is that on an extended culture and single embryo transfer framework, PGTA was cost-effective in women who had 36 years of age or more, regardless of the number of blastocysts, or in women who were 35 years of age or more who had at least 3 blastocysts. However, if the cost of embryo transfers was diminished to half, to around $1,200, which is about the cost that we have here in Colombia, then the threshold for cost effectiveness was pushed towards older age women. And so it was cost effective for women 38 years of age who had at least 4 embryos, blastocysts, or 39 years of age who had at least 3 blastocysts, or 40 years of age who had at least 2 blastocysts. And a similar result was found by Neal and colleagues in 2018. In this case, they used a combined analysis of actual clinical data from 9,000 patients from 74 IVF centers with a mean age of 36 years, who had a median of 4 embryos submitted to comprehensive chromosome analysis. Despite the fact that, as we've seen, eventually the cumulative pregnancy and live birth rates are similar between this PGTA group and the group that only had morphology assessment of embryos prior to transfer, there was a reduction of around $1,000 to $2,500 when there were more than 2 blastocysts available, regardless of women's age. And this was more evident between 2 and 7 blastocysts for analysis. The group with the PGTA achieved a delivery with fewer embryo transfer and had lower miscarriages than the control group. Finally, I would like to talk a little bit about a new technique called non-invasive PGTA. And the reason for the interest in this new technique is that there's no need for embryo biopsy. Embryo biopsies require expensive, high-tech laser equipment and very skilled embryologists in order to perform this microsurgery. So initially, Palini and co-workers described something called as minimally invasive PGT, in which the blastocele liquid from the inner of the blastocyst was aspirated. However, to really call a technique non-invasive, one should not even puncture the blastocyst. So this non-invasive PGTA that is nowadays being studied, it refers to the evaluation of this spent culture media where blastocysts are cultivated. And the one thing is that it has been shown that the DNA content from the blastocyst increases with prolonged culture times. And one has to be very careful, though, not to have DNA contamination, especially from maternal sources. This is a technique that was initially validated in 2016. However, the most recent study by Rubio and colleagues in 2019 has attracted great interest. This pilot prospective blinded study analyzed 150 samples of individually cultured embryos in 10 microliter drops between day 4 to day 5 or day 6 or day 7. The amplification rates were quite good, 95%. And the ploidy concordance between the spent culture media and the trophectoderm biopsy was 79% and 64% for the full karyotype concordance. However, sub-analysis showed that when the culture was extended to day 6 or 7, in other words, more than 48 hours, there was an increase in diploidy concordance to 84% and the amplification rate was 100%. Also, when there was a concordance between the results of the trophectoderm biopsy and the spent culture media, these embryos had an increased implantation rate with lower miscarriage rates. Ongoing studies are still going, and so we are waiting for these results to see if non-invasive PGTA will just be complementary to the regular classic PGTA or whether it would be by itself of clinical use. So we may conclude that PGTA is undergoing steady global expansion. It has evolved greatly since its inception in 1990. Today, it's possible to identify not only normal or abnormal embryos, but to subcategorize them into mosaic or segmental abnormalities, which will avoid throwing away embryos that still have implantation potential. It is very important to understand that there are limitations inherent to the technique because we will never be able to evaluate the whole embryo. Still, there's a group of patients in whom this technique has shown an increased likelihood of positive outcome in a cost-effective manner, as is the case when one is looking for selective transfer of one embryo in order to avoid multiple pregnancy rates, especially in women over the age of 35 who have 2 or more blasts. And finally, time will tell if non-invasive PGTA will be the technology of the future. So thank you again for being with us. I hope you find some of these concepts of help in your clinical practice.